What if I told you that your anxiety isn't a personality flaw, but a biological glitch hidden in a cluster of neurons no bigger than an almond? What if everything you've been told about managing your fear, the breathing exercises, the medication, the therapy sessions, has been treating the symptoms while the root cause sat untouched, deep in the emotional command center of your brain? What if science just found the switch that can turn anxiety off? For decades, we've lived with anxiety, like it's some kind of permanent condition etched into our DNA. Over 360 million people worldwide wake up every day with their chest tight, their mind racing, their body convinced that danger is lurking around every corner. In the United States alone, more than 40 million adults and 4 million children are diagnosed with anxiety disorders. That's not a statistic. That's a silent epidemic. And for most of those people, the solutions have been the same. Manage it, cope with it, learn to live with it. But what if we've been looking at anxiety all wrong? In November 2025, a team of neuroscientists in Spain published a study that flipped everything we thought we knew about anxiety on its head. They didn't just study it, they reversed it. They identified a tiny population of neurons buried deep in the amygdala, the brain's fear and emotion processing center, and they discovered that when these neurons fall out of balance, they trigger anxiety, depression, and social withdrawal. But here's the part that changes everything. When the researchers restored balance to those neurons, the anxiety disappeared. Not gradually, not over months of treatment, instantly. Let me take you inside your brain for a moment. Picture the amygdala as the security system of your mind. It's always scanning, always alert, looking for threats. When it detects danger, real or imagined, it sounds the alarm. Your heart races, your palms sweat, your thoughts spiral. This is the fight or flight response, and it's been keeping humans alive for thousands of years. But in the modern world, where threats aren't saber-toothed tigers but deadlines, social media notifications, and existential dread, that alarm system never shuts off. It's like a smoke detector that goes off every time you toast bread. Annoying, exhausting, and for millions of people, completely debilitating. The amygdala has long been suspected as the source of anxiety, but knowing that the amygdala is involved is like knowing a city has crime without knowing which neighborhood is dangerous. The brain is vast, complex, interconnected. Pinpointing the exact neurons responsible for anxiety has been like trying to find a single broken wire in a city-wide electrical grid. Until now. Dr. Juan Lerma and his team at the Institute for Neurosciences in Spain focused their attention on a gene called GRIC. This gene controls the production of a protein called GLUC, which acts as a receptor on neurons and makes them more excitable. Think of it as the volume knob on a speaker. When GRIC4 is overexpressed, the volume gets cranked up. The neurons become hyperactive. They fire too often, too intensely, and they throw the entire emotional network out of balance. To test this, the researchers used genetically modified mice that overproduced GLUK4. These mice behaved exactly like humans with severe anxiety disorders. They avoided open spaces. They refused to socialize with other mice. They showed signs of depression, including a lack of motivation and persistent hopelessness. In every measurable way, these mice were anxious. But then the scientists did something remarkable. Using advanced genetic tools, they targeted only the neurons in a specific part of the amygdala, called the basolateral amygdala, or BLA for short. They normalized the expression of GRIC4 in those neurons and nothing else. It was surgical, precise, and the results were immediate. The mice that had been cowering in dark corners began exploring open spaces. The mice that had avoided socializing started interacting with unfamiliar mice. The despair-like immobility they showed in behavioral tests, a classic marker of depression, vanished. The anxious behaviors didn't just improve, they were reversed. This wasn't a fluke. The researchers replicated their findings in normal, non-genetically modified mice that naturally showed higher levels of anxiety. They normalized GRIC4 expression in those mice too, and once again, the anxiety disappeared. This validated something profound. The mechanism they discovered isn't exclusive to a lab-created model. It's a general principle of how the brain controls anxiety, and that means it could apply to humans. Let's zoom in even further. Inside the basolateral amygdala, the researchers identified two groups of neurons that work together like a seesaw. On one side, you have regular firing neurons. On the other side, you have late firing neurons. When GRIC4 expression is normal, these two groups are balanced. The regular firing neurons excite the circuit just enough to process emotions without tipping into fear. The late firing neurons keep things in check, preventing overreaction. But when GRIC4 is overexpressed, the balance breaks. The regular firing neurons become too excitable. The late firing neurons go quiet, and suddenly, the entire emotional output of the amygdala is distorted. Fear becomes anxiety. Caution becomes avoidance. Normal stress becomes overwhelming dread. What the scientists discovered is that restoring balance to just the regular firing neurons was enough to fix everything. You didn't need to adjust both groups. You didn't need to rewire the entire brain. One simple adjustment in one specific population of neurons, and the circuit stabilized. The anxiety vanished. This is monumental. For the first time, we're not just guessing at what causes anxiety. We're not treating it with broad, imprecise drugs that flood the entire brain with serotonin or dopamine. We're identifying the exact neurons, the exact gene, the exact mechanism that turns anxiety on and off. It's like finding the breaker box in a house where the lights keep flickering. Flip the right switch, and everything stabilizes. But let's talk about what this means for you. Right now, if you walk into a doctor's office with anxiety, you're likely to walk out with a prescription for an SSRI or a benzodiazepine. These drugs work for some people, but they come with side effects: weight gain, sexual dysfunction, cognitive fog, dependency, and even when they work, they don't fix the root cause. They just turn down the volume on your emotions. They make everything quieter, including the things you want to feel. It's like putting a blanket over a fire alarm instead of putting out the fire. What this research suggests is a future where treatments are targeted. Imagine a therapy that doesn't alter your entire brain chemistry, but instead adjusts the excitability of a few thousand neurons in your amygdala. No cognitive impairment, no emotional numbness, just a restoration of balance, just your brain functioning the way it's supposed to. Now, let's address the elephant in the room. This study was done in mice. Mice are not humans. Their brains are simpler, their behaviors are easier to measure, and their genetics are easier to manipulate. But mice and humans share the same basic brain structures. The amygdala in a mouse works the same way it does in a human. The GRIC4 gene exists in humans. The gluK4 protein exists in humans. The regular firing and late firing neurons exist in humans. The circuitry is conserved across species. So while we can't say with absolute certainty that this mechanism applies to every human with anxiety, the evidence strongly suggests it does. In fact, researchers have already started looking at GRIC4 expression in humans. Preliminary studies show that people with anxiety disorders often have variations in the GRIC4 gene. Some have mutations that increase its expression. Others have epigenetic changes, environmental factors that turn the gene on or off, linked to early life stress, trauma, and chronic anxiety. The genetic fingerprint is there, and if the fingerprint is there, the treatment pathway might be too. But there's more to this story. The researchers noticed something else in their experiments. While the anxiety and social behaviors improved dramatically, some cognitive functions didn't fully recover. Specifically, the mice still struggled with object recognition memory tasks. This suggests that anxiety doesn't just live in the amygdala, it spreads. It affects other regions of the brain, like the hippocampus, which is critical for memory and spatial navigation. Anxiety isn't just an emotional disorder, it's a cognitive one. It changes the way you think, the way you remember, the way you perceive the world. This is why anxiety feels so totalizing. It's not just that you're scared, it's that your entire reality is filtered through fear. You can't trust your thoughts because your thoughts are shaped by the distortion in your amygdala. You can't trust your memories because your memories are encoded under stress. You can't trust your perceptions because your perceptions are biased toward threat detection. Anxiety doesn't just make you feel bad, it makes you see the world as a bad place. So what do we do with this information? How do we take a study done in mice and turn it into something that helps the millions of people suffering right now? The answer is twofold: technology and awareness. On the technology side, scientists are already working on developing drugs that target GRIK4 expression specifically. They're exploring gene therapies that could normalize the activity of neurons in the basolateral amygdala without affecting the rest of the brain. They're investigating neuromodulation techniques, like transcranial magnetic stimulation, that could adjust the excitability of neurons non-invasively. But technology is only half the battle. The other half is awareness. For too long, we've treated anxiety as a character weakness. We've told people to just relax, just breathe, just think positive. We've pathologized fear and medicalized worry, and in doing so, we've missed the bigger picture. Anxiety is not a failure of willpower. It's a failure of neuronal balance. It's not about being strong enough to fight it. It's about restoring equilibrium in a system that's been knocked off course. And here's the most important part. If anxiety can be reversed in mice, it can be reversed in humans. This isn't speculation. This is neuroscience. This is reproducible, measurable, observable. The path forward is clear. It's just a matter of time, research, and investment. The question isn't if we can fix anxiety, it's when. Imagine a world where anxiety is treated not as a lifelong condition, but as a temporary imbalance, where a diagnosis doesn't mean decades of medication, but a targeted intervention that restores your brain's natural equilibrium, where you don't have to choose between feeling numb on drugs or overwhelmed without them, where your amygdala works for you, not against you. That world is closer than you think. So the next time your chest tightens, your thoughts race, your body tenses up for no reason, remember this: it's not your fault. It's not a flaw in your character. It's a circuit in your brain that's out of balance. And balance, as we now know, can be restored. If you found this fascinating, subscribe to the channel, hit the like button, and drop a comment below with your city. Let's build a global conversation about the future of mental health.